This is going to be the first video in Unit 3 about the cells and their functions. This is this video about the microscopes, the two different microscopes, the light microscope, as well as about the electron microscope. The first person who we have to talk about is Robert Hooke, a German scientist who lived in the 1600s, and he made the first microscope around 1665. He was the one who used the cell as the name first. He used that for the structures he observed in the specimen of the cork. These small kind of small chambers he referred to as cells of the monks, for example, but these are technically cells all around. And he reconnect the name cell to him. That was his microscope. This was a simple microscope with one lens only. Do we really see everything? That was one of my favorite people in biology is Anthony von Leeuwenhoek. He was a Dutch merchant, a fabric merchant. He had no high level of education. He got the business from his father, and he was so eager to make the finest fabric in whole Europe. So he started to polish some glasses to be able to see closer, so magnify the fabric, and he would be able to count the thread count. He made also at the simultaneous time with Robert Hooke his microscope, and he was also a very curious person. He viewed bacteria, the plague from his teeth. He checked pond water, and he found protists in the pond water. He checked his semen for sperm cells, and he didn't know at that time what those were. He also checked the blood and saw his blood cells. His instrument, so he was really a curious person. This was his microscope. It was a plate, and the lens, the polished lens, was mounted in that brass plate. The specimen he wanted to check was placed on the needle in here, and with the two screws, one of them here and one of them is here, and also here, he was able to focus the specimen under the lens, so he had a sharp image. He published his first observations in 1674, a little bit later after Robert Hooke. That's questionable whether he was inspired by Robert Hooke or by himself. There's no evidence that they worked together, pretty much. He was a Dutch merchant and Robert Hooke was an Englishman, but he was really just did the microscoping as his hobby. We call him the father of microbiology because he published so many observations, and he really gave us a very detailed descriptions of what he saw under his simple microscope. Now, we use a light microscope today, and the microscope what we are going to use in the class, it's a compound microscope. Compound because it has two lenses, the ocular piece and the objective piece. These are the two pieces. The ocular lens usually has 10 magnification, 10 times, and the objectives can be 3 times, 10 times magnification, or 40 times magnification. How to figure out the total magnification? Magnification equals to the ocular pieces, ocular times objective. So if I have the 3 times magnification at objective and 10 times the ocular piece, the total magnification for that is 30 magnification. And here it's 100 magnification, and for that it's 400 magnification. How simple to figure it out. Way back to Robert Hooke and Anthony von Leeuwenhoek, they used only simple microscopes. They had only one out of the 2 lenses. The microscope, we have to talk about the magnification and resolution. That is really important, not only for the light microscopes that they use light to go through the specimen and be able to see under the microscope, but as well as for the electron microscope. What is magnification? Magnification is how big the object, how much the apparent size, increasing the apparent size. For example, here's the dog, the magnification of the nose. This is magnification from the dog towards the nose. See how much more we see the nose. That's the magnification, how big we will see it. On the other hand, the resolution means clarity of an object, the ability to show 2 nearby objects as separable and as separate from each other. In here, you really cannot see the resolution as much as on that picture. You can distinguish between this indentation and this indentation too, but on here you cannot see anything else. On the second picture too, you can see the individual hair particles as well as in here. If we compare it, you cannot see it. So the resolution of the first section is bigger resolution compared to the second. The magnification is both can do the same magnification. The increases the apparent size, but not the resolution. Later on, it will be really important. For changing the light microscope to another type of microscope, such as electron microscope, was a big change. The light microscope had a chance to do the magnification by the most as 1,000 times, but the electron microscope can do the magnification 1,000,000 of times of magnification we can get. So this is really, really great. Electron microscope uses electron beams of electrons to go through the specimen in here and make a image here at the bottom of the electron microscope. It's huge, so it is 2-3 yards tall machine, so you really cannot just take it back and forth. The electron microscope comes in 2 different types. The scanning electron microscope. The scanning electron microscope, like in its name, does the scanning of the surface. On this picture you see a virus, how the surface was covered with very thin layer of gold, and as the electron passes through the specimen, it will make an image of the specimen. This scanning is pretty much gives us a 3-dimensional picture of the specimen. On the other hand, the transmission microscope is kind of going through, not only just checking the surface like in the scanning, but goes through the specimen. You have to slice the specimen into very, very tiny, thin slices like a salami, and you have to stain the specimen with heavy metal atoms to make an image on the camera. With a transmission microscope, you will get a detailed internal structure. This is one of my favorite organelles, the mitochondrion. You can see the two membrane structure, the internal and outer membrane structure. So it gives us a detailed internal structure. On the other hand, the scanning is on the surface. Both cases, both the scanning and the transmission microscope, you have to kill the specimen. You have to apply either gold cover for scanning as well as heavy metals as you slice the specimen up. The electron microscopes give us a great magnification. This is the end of the first video about the microscopes.